Hello everyone, here is Julia from Rey Juan Carlos University. Welcome to the Journal of Ecology video podcast about the paper Evidence for a Stochastic Geometry of Biodiversity, the Effects of Species Abundance, Richness, and Intraspecific Clustering. Despite the weak consensus regarding biodiversity theories, McGill 2010 identified a common set of three integrative rules which are shared implicitly or explicitly in all biodiversity theories. These three rules describe what has been called a stochastic geometry of biodiversity. The rules described by McGill also represent a different starting point for testing neutral predictions based on statistical arguments. The rules assume that, first, most plant species show intraspecific clustering or aggregation. That means that most individuals have another conspecific individual as neighbor much more frequently than if they were randomly placed. This pattern could be a response to dispersal limitations or to microhabitat preferences. Second, the abundance of a species in a community is following a log-normal distribution. That means that communities are characterized by many rare species and only few common species. And the third rule is called the independence species arrangement and proposes that in species-rich communities, most interspecific individuals are located independently in space with respect to each other. So, in species-rich communities, the frequency of interspecific spatial associations is lower than in species-poor communities. The first two rules, or hypotheses, have received strong support from empirical studies and are known to have important implications for the spatial organization of plant communities at fine scales. However, the independence arrangement hypothesis that proposes independence placement of species with respect to each other in species-rich settings represents the less studied rule and also the most controversial. Previous studies testing the independence arrangement hypothesis are really scarce. These studies explored the full array of species associations, that is, all the possible species pairs in species-rich communities, giving empirical support to the rule. However, these studies have only reported the effects of species richness at the plot level by comparing across sites or plots with different richness levels. In addition to the effects of species richness on the likelihood of two species to encounter and associate, intraspecific clustering and the log-normal distribution of species abundances might also influence the likelihood of two species to encounter and interact. For example, species with a strong intraspecific clustering are surrounded by more conspecific individuals than heterospecific individuals, which reduces the likelihood of interspecific associations. And in communities where there are many rare and only few common species, the presence of many species at low densities may also reduce the likelihood of two species encountering each other. In addition to that, we know that the species richness varies with a scale within sites as the distance increases from an individual focal species. Thus, it is necessary to test the effects of species richness as a potential driver of independence across scales, as well as the potential effects of the relative species abundances and intraspecific clusterings as other potential drivers of independence arrangement of the independent species arrangement hypothesis. In this context, we wanted to assess the importance or prevalence of the independence species arrangement hypothesis, and we were really interested in its potential drivers, but specifically at different scales as the distance from an individual species increases. To assess the importance of the independent species arrangement hypothesis, we employed spatial point pattern techniques together with specific new models that controls the effects of environmental heterogeneity. So, we analyzed all the pairwise spatial associations between species in a fully mapped community to test whether spatially independent placement prevails between species. So, we established a permanent plot in a very species-rich Mediterranean shrubland in Spain. In this plot, all the individuals of all the perennial species were located by their x and y coordinates. In total, we located about 8,000 individuals of 48 perennial species into a 60 meters square plot. Specifically, we asked whether interspecific independent patterns prevail, which types of departures from independence appear in the plot and whether independence was related to intraspecific clustering, relative abundance, or species richness. And of course, the scales at which that happens. So, does interspecific independent pattern prevail in the plot? The answer is yes. Okay, here in purple I'm showing you the proportion of species pairs being independent at different scales as we increase the distance from every target species. As you can see, most species pairs, around 80%, were independent at all the scales analyzed. And this effect was especially strong at scales lower than 50 centimeters from a target species. And which types of departures from independence appear in the plot? Okay, only around 20% of species pairs show deviations from independence, specifically following a pattern of segregation at scales higher than 50 centimeters. We tested whether the likelihood of a pair of species being independent at a particular scale was related to the relative abundance of the species in the plot, to the intraspecific clustering of the species at a particular scale, and or to the species richness experienced by a target species at this particular scale. Here I'm showing you the coefficients of these three explanatory variables in a regression, in dark colors when the variables were significant and in light colors when they were non-significant. As you can see, species richness in red had positive coefficients that were significant from 10 centimeters. That means that the more richness a species have in its surroundings, the more likely to be independent of other species. We found the same pattern for interspecific clustering. So in blue in the figure, a species surrounded by conspecific individuals is more likely to be independent of other species. In black, I'm showing the coefficients for relative abundance. In this case, were negative, but significant at all scales. That means that the more abundant a species is in the plot, the less likely to be independent. In other words, the more abundant, the more likely to establish associations with other species. So, in conclusion, our findings agree with the independent spatial arrangement between species, thereby providing empirical support to a stochastic geometry of biodiversity. And our results also show that the number of pairwise independent associations that a given species can establish is related to the relative abundance of this species, to the intraspecific clustering pattern of this species at a given scale, and to the species richness found in the neighborhood of this particular species at a given scale. There is much more information in the paper, so if you are interested in reading it, you can find it in the Journal of Ecology webpage. Thank you very much for listening. If you have any comments or questions, just send me an email and I'll answer. Thank you. Bye bye.